has been served as a site peer mentor and has conducted research in my neuroscience research lab. After graduation, she plans to continue working at the hospital and prepping for the MCAT. So today, Julie is going to be talking to you about her research project involving overglycation and mTOR. So please join me in welcoming Julia Johns. Well, thank you, Dr. Franklin, and good morning, everybody. My name is Julia, and I'm so excited to present to you all my capstone today, entitled Examining the Effect of Increased Overglycation on mTOR and Phosphorylated mTOR in OGA Knockdown Mice. So, just an outline of my presentation, I'm going to start off with talking about overglycation and mTOR and phosphorylated mTOR. So what is it, why is it important, and why is it worth studying? That will lead me to my aims and methods, and then my anticipated outcomes, and I'll finish by talking about future studies. So, what is overglycation? The first thing that I need to talk about is the hexosamine biosynthetic pathway, which I will be referring to for the rest of the presentation as the HBP. So, hexosamines are commonly used in the glycosylation for the modification of nucleic acids, proteins, and lipids. And so, overglycation is facilitated by and is dependent upon the synthesis of unique-gluconate, which is the byproduct of the hexosamine biosynthetic pathway. As you can see, there are multiple converging pathways, enzymes, and subsequent reactions in this pathway, including glycolysis, the fatty acid oxidation and TCA cycle, and pyrimidine biosynthesis, which makes this pathway very complex, and therefore it is very important to study. So, what is overglycation? Overglycation is a post-translational protein modification with O-linked N-acetylglucosamine, or O-gluconate, on serine and threonine residues. It's involved in gene regulation, development, signal transductions in the cell cycle, and operates through two cycling enzymes, OGA and OGT, which makes it a cyclic and reversible process. So let's say I have a target protein of overglycation, and I want to add on an O-gluconate variety. OGT, or O-gluconate transferase, is responsible for this activity, and with the hydrolysis of the glycosidic linkages, OGA, or O-gluconate ACE, will remove the O-gluconate variety off of that protein. So, overglycation and its dysregulation has been implicated in multiple diseases, including liver cancer, type 2 diabetes, lung cancer, atherosclerosis and heart failure, gastric cancer, Alzheimer's disease, and Parkinson's disease. And a common trend that I found within my research was that through all of these diseases, overglycation was actually upregulated. And so that kind of triggered my research in OGA knockdown for increased overglycation. So, scientists have suggested a cross-communication between overglycation and phosphorylation due to the common protein target residues. So, the serine and threonine residues are targeted both by overglycation and phosphorylation. However, what's different is that O-gluconate varieties are added onto the target protein in overglycation, whereas in phosphorylation, we're focusing on the addition and removal of a phosphate group. They're also involved in different things. Phosphorylation is involved in cell signaling, metabolism, cell cycle regulation, whereas O-gluconate that is more involved in nutrient sensing, gene regulation, and the stress response. Something else that differs is that O-gluconate we utilize UDP-gluconate as the substrate. So going back to the HBP, that was the product of the HBP, and so O-gluconate does utilize this as the substrate, whereas phosphorylation is going to utilize ATP as a phosphate domain. So this leads me to mTOR and phosphorylated mTOR. So what is mTOR? mTOR is the mechanistic target of rapamycin, which originated from Streptomyces hygroscopicus, which was actually found in a soil sample off of Easter Island in 1964, in the efforts to find antimicrobial agents. So, mTOR is a serine and threonine kinase, and it regulates cell growth and metabolism, and it also displays antifungal, immunosuppressive, and anti-tumor properties. So, mTOR is special in the way that it operates. It operates through two complexes, mTORC1 and mTORC2. As you can see over here, this is mTORC1, and this is mTORC2. And so mTORC1 is actually smaller and has about 6 subunits, whereas mTORC2 has 7. So mTORC1 is sensitive to rapamycin inhibition, whereas mTORC2 is only susceptible to rapamycin inhibition through long-term exposure. So mTOR's role in human disease. mTOR dysregulation has been observed in approximately 30% of cancers, and an increase in mTOR activity has been shown in the progression of diabetes, high insulin levels, high glucose tolerance, and beta cell decline. It has also shown critical roles in muscle function and health. So it promotes muscle growth, but it can also lead to muscle atrophy if it is chronically activated. So mTOR1 and mTORC2, they are both implicated in neural development and neural circuit formation. However, the biochemical function between the brain and both of these mTOR complexes are mostly unknown. So, phosphorylated mTOR. You might be asking, what's different between mTOR and phosphorylated mTOR? So, phosphorylated mTOR is just the activation of mTOR by phosphorylation at serine 2448. And pMTOR expression has been reported in multiple cancers and the upregulation of cell growth and metabolism. However, the expression and activity of phosphorylated mTOR in normal human tissues has yet to be investigated. So, what are some connections that we can draw between methylation and mTOR activation? The first thing is that both of these modifications are major nutrient sensors. So, therefore, these pathways and their functions can interact and therefore be modulated. So mTORC1 is regulated by glutamate, it affects OGT expression, and it regulates cell growth and nutrient excess. It is also shown that OGA knockdown and the administration of OGA inhibitors increases the activation of the mTOR signaling pathway. Another thing to note is in an OGT knockdown, this would suppress mTORC1 activity, whereas the total inhibition of mTOR will increase OGA, which would decrease O-glutamylation overall. However, despite all of these findings, not one study has proved that O-glutamylation directly targets the mTOR pathway. So while there are connections, there's not a specific study that has proved this connection, which is going to lead me to my aims and methods. So the first thing that I want to talk about is the TET-on and TET-off system. So what is this? It's a manipulation of genes for the administration of tetracycline or its derivative doxycycline. So in the presence of doxycycline, the TET system would be on, therefore inducing gene expression. If the TET system is off, the gene expression would not be induced due to the absence of doxycycline. So here is the TET-on and the TET-off system. And as you can see, in the absence of doxycycline binding to this promoter, there is no gene expression. However, in the TET-on, the presence and the binding of doxycycline will actually induce the gene expression. So all of my aims and methods will be carried out using the Western blotting process. So what is a Western blot? Western blot is a simple gel electrophoresis. So my wells will be filled with my samples and run through the gel. Once this gel is run, it's the assembly of a sandwich. So this is kind of a fun way to think about it. You would start with a sponge, and then we have a piece of filter paper, and then on goes the gel. Next, the PVDF membrane, which is going to be a very important part of the Western blot process. Another piece of filter paper, another sponge, and then it can successfully run through the transfer. After successful transfer onto the PVDF membrane, this membrane will go through a series of washes. And the two washes that I really want to focus on today are the washes with the secondary and primary antibodies. So the primary antibodies that will be used in this Western blotting process are mTOR and phospho-mTOR. And these will be mixed into a 5% milk solution and washed with on this shaker with agitation. And then the secondary antibody is going to be goat anti-mouse because the subjects that are in my study are going to be mice. Next, this PVDF membrane would go through imaging, and that is how we would obtain results. So I'm going to introduce my groups because they're going to stay consistent throughout specific aim 1 and specific aim 2. So in group 1, it's going to consist of 6 wild-type male mice, 3 dogs on and 3 dogs off. These mice will be sacrificed, their brains will be harvested and dissected. However, the samples that would be used in this study are previously homogenized and prepared. In group 2, it will be 6 knockdown male mice, 3 dogs on and 3 dogs off. And similar to group 1, these mice will be sacrificed, their brains will be harvested and dissected, and the samples for this are also previously prepared and homogenized. Which is going to lead me to my specific aim 1. Specific aim 1 is to examine the effects of increased O-glutamylation on total mTOR levels in wild-type and OGA knockdown mice by utilization of the TET-on TET-off system. My hypothesis is that due to no genetic modifications in the wild-type mouse, the presence or absence of doxycycline will have no effect on O-glutamylation and total levels of mTOR. However, in the knockdown mouse, total levels of mTOR will increase due to increased gluconylation. So my expected outcomes for specific A1 group 1 is that since both of these groups are wild type, the dox on and dox off, the presence and absence of doxycycline, is not actually going to affect OGA expression because there are no genetic modifications. So then I'm going to expect normal O-glucose cycling and no change in mTOR levels. However, in my knockdown mice, the presence or absence of doxycycline will induce the OGA knockdown, which will lead to increased gluconylation levels and an increase in mTOR levels. My specific aim 2 is to examine the effects of increased gluconylation on phosphorylated mTOR in wild-type and OGA knockdown mice by the utilization of the type 1 tag-off system. My hypothesis is that in the wild-type mice, both dox on and dox off, there will be no change in phosphorylated mTOR levels. However, in the knockdown mice, I'm hypothesizing that due to increased gluconylation, the phosphorylated mTOR levels will decrease. So my expected outcomes for specific A2, group 1 is going to be very similar to specific A1 group 1 in the fact that there are no genetic modifications, which will lead to normal glucose cycling and no change in the phosphorylated mTOR levels. However, for specific into group 2 in the knockdown mice, this is where things start to get a little bit different. So in the presence or absence of doxycycline, the OGA knockdown will be induced, which will increase gluconylation levels overall. And I'm hypothesizing a decrease in mTOR levels. The reason why I am hypothesizing this is because I want to go back to what I talked about with the crosstalk between gluconylation and phosphorylation. My hypothesis is that if this protein, mTOR, is already phosphorylated, could there be a chance that this protein can even be gluconylated? And so therefore, I'm going to say that it's not, and that's my hypothesis. And so if it cannot be gluconylated, then the levels would decrease. So for future studies, obviously there's a critical need for research on the mechanisms in which gluconylation, phosphorylation, and mTOR collaborate. An idea that I really wanted to do, but I didn't have time to do it, was to investigate inhibitory effects on the rate-limiting steps of one pathway, for example, the HVP, and its effects on another, for example, glycolysis, to introduce new ideas about inhibitors as treatment for disease. So there are inhibitors of certain enzymes within these pathways that do exist. However, it hasn't really been investigated what that's doing to the other pathway. And lastly, HVP gluconylation in both of the mTOR-C complexes and its activation, and focusing on where these pathways intersect and determining how the dysregulation of one can lead to the dysregulation of another, and therefore lead to human disease and disease progression and pathogenesis. So my acknowledgements, I want to thank God for giving me the opportunity to study at SEU, my family and friends for always supporting me, my professors for teaching me pretty much everything that I know, and for Dr. Breitland for the amazing opportunity to be a part of this research. And here are my references. I would like to open to any questions. We have time for a couple of questions. Yes. In your electrophoresis, what are you, maybe you said this, but what are you putting in the wells? Like, what's the actual example? Good question. So in my wells, we can go back to the question here. So this gel consists of about 10 to 12 wells. And in the well, so in the second well, I'm going to put the molecular weight ladder. This is going to help whenever my samples are actually run, determining where these proteins will actually show up based on molecular weight. So for mTOR and phosphorylated mTOR, I'm expecting that to show up around 289 kilodaltons, which is going to show up towards the, up on the block rather than down on the gel electrophoresis. So in the samples, it's going to consist of the brain homogenate, frontal cortex homogenate, sample buffer, and then water. And it's about 20 microliters in each well. Yes?
You could pull your slide on the difference between gluconylation and what I can't say this word, Dr. Chang. Gluconylation and phosphorylation. So I think if I followed this correctly, you have two different enzymes involved in your gluconylation and phosphorylation. So given that there are two different enzymes on the target, right, whenever you're doing something to affect the processes, why assume that if gluconylation is affected, phosphorylation follows? considering that there are two different enzymes involved in the process?
Okay, that's a good question. Honestly, the mechanisms between these two processes haven't really been studied. And so I'm going to be honest, I don't know exactly the ways of those mechanisms and how they collaborate. However, my hypothesis just came from that because these both occur on serine and threonine residues, if the residue is already phosphorylated, which the antibody in the lab is on serine 2448, so the one that I mentioned with phosphorylated mTOR, that if it's already phosphorylated, there wouldn't be room for that loop-back moiety to be added onto that same residue. So for the residues, we don't know exactly where the loop-back moiety is added onto.
Yeah,
Is
that's
that
going to
answer
jump
your
to the
question?
next question is, if you're not having a target moiety, if you're moiety, how are you going to be able to actually really know that that is affected in the process? So, I mean, I understand where you're coming because you've not done the whole deal, but I think that's the flow of the thought, right? This is what's going to happen.
So kind of figuring out what specific residues of glutamylation and phosphorylation would be occurring is definitely something that would need to be looked into. Great job, Julie. Thank you. Yes, and then we can say we have effectively preserved our beta cell population. Now, for future studies, first I'd like to say this disease is progressive, as I mentioned, and so if we can develop earlier detection methods and get to a diagnosis faster, our treatments would be more effective. Whether that's this thymosin alpha treatment or any other treatment that comes along, the faster we get to it, the faster we can delay disease progression and more efficiently treat the problem. And then, shockingly, research has shown us that beta cells actually have the capacity to regenerate on their own. And so if we can stop this immune attack on our beta cells, protect them maybe with this hormone, we can give them the space to regenerate on their own. And we can repopulate that population of beta cells, leading to an increase in insulin and maybe one day even curing type 1 diabetes. And so it's important to note I am in no way proposing a cure as of right now for type 1 diabetes. This is more a novel therapeutic approach that takes what we have right now and also adds the addition of addressing the underlying immune dysfunction. We're expecting to delay disease progression and preserve the function of the cells that we have left, as I said, to hopefully increase patients' lifespan and decrease the risk of some of those long-term complications that I mentioned. And then, as I said, also by refining our diagnostic tools, we're going to enhance the efficacy of any treatment that comes along, especially at thymosin alpha treatment. And so for my acknowledgments, I'd first like to thank God for the opportunity to be here today and to study at Southeast University. It's such a blessing. I'd like to thank my family and my friends. Your support and encouragement have gotten me through these 3 years, and it has just been amazing. And then my advisor, Dr. Shaw, your wisdom and your patience with me has been invaluable, and I'm so grateful to you and to all the professors here. You guys have all made a lasting impact on my education, but my life as well. So here are my references, and I'd love to take any questions that you have.
So you mentioned you're going to measure the cytotoxic T cells that recognize the beta cells. How exactly are you going to do that?
So that's where my staining procedure will come in. Hopefully, we will, at the end of the first methodologies for AIM-1, we will stain the cells in the petri dish with different types of stains that will show us have we changed the concentration at all from our control group. So we'll stain the control group, we'll see how many cytotoxic cells and how many regulatory cells we have, and then we'll stain the experimental group and see if there has been any change between that and the control groups.
So you showed us this is a common therapy for lots of diseases. Is there a reason it hasn't been used in diabetes before? Is there a negative side effect? If it's been used for so many things, it's interesting to me that they haven't thought of using it for diabetes.
Yeah. I mean, I can't speak for everyone and why they haven't thought of that maybe necessarily, but in the context of every other disease that I mentioned and that I looked into there, have been no negative side effects. Because this is a hormone that is produced by our body, it works amazing because we already have it in our bodies, and so we're not rejecting that at all or having any side effects. It is just an immune modulator. It's in the context of different diseases, it manipulates different things. And so in some of those different diseases, it would maybe increase our regulatory cells or decrease our cytokine productions. It looks a little bit different in each disease. I'm not sure why it hasn't been studied at type 1 diabetes. It's being explored in the context of other autoimmune diseases I know right now. But I don't know. I don't expect there to be any negative side effects, and so I don't think that's the reason. Maybe just a lack of progression in research of this disease. Okay. Yes.
Yes. Can you pull your methods where you have the time set for one's concentration? I'm not sure if I'm following it correctly. I want to make sure I think it was 200 microper per kilogram per body. So that's what you are injecting, or that's what's in the body already as a normal mass?
So that's what I'm injecting.
That's what you inject.
Yes.
Oh, that's it. I wasn't sure. Okay, what's the basis of that? Because I think that's what now goes into the question of why are you not using this, right? For one, as you have indicated very early on, it's a very general regulator for many different things in the body. So messing up with the concentration of that can mess up a lot of processes, right? So what is the basis of the 200 microgram, and what is the normal amount usually?
So I'll answer that question in two parts. The normal concentration of this hormone in the human body, I can't give you an exact number because it looks different as we age. And so as we reach the age of puberty, these levels decrease. So it's going to look different day to day and progressively as we get older. The basis of the 200 micrograms per kilogram really just comes from my exploration of this use in the context of other diseases. This was kind of the standard for the preclinical trials in mice models. And so that's really where I adapted it from. But I think that if we were able to discover that this worked, maybe in a petri dish specific A1, then we could manipulate different dosages to see what effects we have. Because you're right, you don't want to mess up anything else in the body. Everything is very fine-tuned. But the basis of that number was really just a preclinical trial based off other disease contexts. Yes.
So this talk, by the way, I think that research is very interesting. My question to you is, so the tyrosine or alpha-1 is going to be injected specifically targeting CD8 and CD4 cells, right? How is that going to be because you were talking about this being used in like a clinical trial potentially in the future. How are you going to be able to isolate that it's only effect with the cytotoxic T cells effect on the pancreas, on the pancreas? When you talk about downregulation of CD4, that sounds like HIV, right? But you don't want that. And so how are you going to be able to isolate, like just thinking in the future, how would you think to isolate just affecting the pancreas and downregulating the immune response rather than suspending immune response?
That's a great question. I think that this is where the difference in my two aims comes in. And so it's important to establish that first aim where we are isolating it in a petri dish, because you're not isolating your cells in the body, right? When you're doing an injection, like you said, it's going to affect your entire body as a system. And so if we can evaluate the effects in a petri dish when we have just isolated those CD4 and the CD8 cells, I think that will help us. It will help give us a baseline with that dosage, like Dr. Corrin was mentioning too, that will help us know where to start when looking at the full body, because we don't want to induce things like HIV or other things and stuff. If we can establish that this works in the context of CD4 and CD8 cells, then we can maybe start looking at other cells. Maybe in a petri dish, we can isolate other cells and see what the effect would be before we inject it into an entire system like the human body or even a mouse. Did I answer your question?
Yeah. Unless things are fingered. You. is.
Or video.
Do.
He's rated up my Capstone by color. Sorry, it's the full is best everything. Forget to Guess I can hear. Hi everyone. I have the great honor of introducing Portland Williams. She is majoring in biology with a concentration in medical science. She is from Ocala, Florida, and has been here for 4 years. While at SNU, she has been a member of the PHSO and served as a TA for Biology 1 and 2 for 3 years. After graduation, she plans to take a gap year, and during that time, she plans to work as a medical assistant and then apply for med school and hopefully start the following year. Thank you for background and studying interest in me today. Hello everyone, my name is Cora Williams. Today I'm going to be presenting my capsule topic of investigate the inhibition of DDR6 solution hormone and its receptor pathway in pregnant moms that can be a target for reducing nausea and vomiting during pregnancy. So nausea, vomiting and pregnancy is one of the most common and challenging pregnancy complications with activity known as morning sickness, and that's been found effective 80% of women during pregnancy. And looking at the United States, it has found that 4 million women each year experience these type of symptoms. And roughly about 1.7 billion per year has been on a population due to this complication. So when a pregnant woman is experiencing nausea vomiting during pregnancy, it typically begins during the first trimester of pregnancy. And so it will begin the period of 2 to 4 weeks after fertilization, and then the symptom will begin to peak around 9 to 16 weeks of pregnancy, and generally coming in around 24 weeks, but this all depends on how severe the symptoms are. So the severity of morning symptoms can range from mild, moderate, to severe, depending on how intense the symptoms are. So most severe form of morning sickness is known as hyperemesis gravidarum, which has been the cause of most hospitalization during the first half of pregnancy, and also the second cause hospitalization for a full-term pregnancy. So when a pregnant woman experiences this severe illness, it has been thought that they experience excessive vomiting that causes dehydration, ketone urine, and about 5% loss and body mass. And with women who have this type of severeness illness, it has thought that they have prolonged symptoms that can last up to labor and even birth, to where having been working to terminate their pregnancy. So current treatments that are given to treat nausea and vomiting during pregnancy do not attack the biological cause of it, but are just given to relieve the symptom that they are experiencing. So type of medication that are given are vitamin B6 supplements and pharmacological drugs such as antihistamines and corticosteroids and antihistamines. And while the medications are given, they have been found that they do an excellent job at relieving the symptom that they are feeling, causing something to still experience the symptom that they are still. And also, it's little evidence that has been shown overall and what type of impact it's going to have on the fetus. So now I'm going to look at the biological effects it has on a mother. So it causes a decreased immunity due to the nausea that they're feeling, because they lose their appetite. And it also causes a decrease in their weight due to the excessive vomiting that they're experiencing. And then lastly, it also puts them at a higher risk of complications such as severe pelvic hypertension, proteinuria, which is protein found in the urine, and preeclampsia. And not only does it have biological effects, it also has psychosocial effects such as increased feeling of depression. It decreases their overall quality of life, which endangers their well-being and work life. And it also decreases their relationship with their family members. So it was additionally that cause of morning sickness was actually due to the hormone known as human gonadotropin, due to the relationship in time when this hormone starts to be produced and when nausea and vomiting symptoms begin to develop. But recently in 2024, that we discovered that the actual cause of morning sickness was due to the hormone known as GDF15. And so now, knowing that the overall cause of morning sickness is due to this hormone, we can develop a treatment that inhibits its effects. So you may be asking, what is GDF15? So GDF15, known as growth differentiation factor 15,
It's a stress-induced cytokine hormone that signals the receptor GFRAL, which stands for glial neuroreceptor factor receptor alpha-like, and it is part of the transforming growth factor superfamily. And so with this hormone, it plays a role in a person's overall appetite, their body, their skeletal muscle mass, and their body mass. So it's produced by many tissues such as organ acids, the digestive system organs, and in males, it's produced by the prostate, and in females, and a non-pregnant female, it's produced by the endometrium. But when a pregnant woman is pregnant, when a person is pregnant, it's produced mainly by the placenta. So looking at this graph, you can see the overall role that GF plays during a body, during a pregnant one. As we already know, with high levels of it, it causes nausea and vomiting in the more severe form of it. It also has been found that it causes increased levels of maternal sugar levels that can lead to gestational diabetes. And not only does it have these good impacts, it also has it not only does it have a bad impact, it also has good impact, such as maintaining the level of the placenta, while also putting at increased risk of having a miscarriage. So now that we know the overall role what they play in the body, now we need to look at the mechanism that what leads to causing nausea and vomiting. So first it's going to be produced by the placenta, where it is going into the maternal bloodstream, by the word, going to walk to the brain, binding its receptor in the brainstem, and this then will lead to the activation of its core receptor known as RET. And this activation of RET would then lead to activation of many different signaling pathways that will then lead to activation of different biologics such as the adipocytes, the liver, and the stomach, where they will complete tasks result in nausea and vomiting symptoms. Now looking at the receptor, it's a receptor in the glial neurotrophic factor family receptor alpha-like. It's a heterodimeric receptor that forms a complex receptor with its core receptor known as RET, which stands for rearranged during transfection. So this receptor is found in the brainstem where it has no blood-brain barrier function, allowing GF15 to go directly into the brain and bind with its receptor. And so now that we know the overall role that GDF15 plays in the body, we can develop a therapy approach that inhibits this that could lead to a decrease in nausea and vomiting during pregnancy. So this was a figure that was taken from a study that developed different therapeutic approaches in inhibiting GDF15. So first you can have an antibody that binds directly to a receptor, preventing it from binding with GDF15. Then you can use a small peptide antagonist to where one GF13 binds to the receptor, the small peptide antagonist is going to bind to it, preventing it from activating the core receptor RET. And then you can use a small molecule where instead of preventing GDF from binding to the receptor, stimulating the activation pathway. For this study, you will be using a small molecule such as an antibody that's going to bind to GDF15, preventing it from binding to the receptor. So the antibody that I'm using is a molecular antibody that has punchgromad that is a potent inhibitor of GDF15. It has been used in many cancer trials, such as the cancer known as cachexia in mice, and even in phase 1 trials for humans. And in these studies, you have found that once punchgromad has been injected, it binds to GDF15 in the bloodstream, preventing it from binding with receptor. So after it has been shown that once punchgromad was injected into these individuals, there was an increase in their survival rate in the mice and also in the cancer remission for the humans. So this then led me to my menstrual question. Could punchgromad inhibit activity of GDF15 cause an increase in the sense of nausea and vomiting during pregnancy? So my specific one is to determine the effect of inhibiting GDF15 using a monoclonal antibody known as punchgromad on reducing nausea and vomiting-like symptoms in pregnant mice. And so I hypothesized that if GDF15 activity is utilized in the maternal blood by injection of punchgromad in pregnant mice with high levels of GDF15, this would then decrease the nausea and vomiting symptoms that the pregnant mice are experiencing. So my experiment design for it is that I'm going to have 4 weeks where I'm going to have 3 experimental groups and 1 control group. My experiment is going to be a low dose group, a medium dose group, and a high dose group. With each dose group receiving a different amount of the punchgromad. And then my control group then received no treatment out. And so after they are randomly assigned to their group, where each group containing 8 mice, they're going to receive a subcutaneous injection every 48 hours. And my timeline for this will be for about 9 days, which will begin in the mice at gestational day 6 to 14 of pregnancy. And this stimulation will elevate with levels of GDF that you will begin to elevate. And then after that, how I plan to collect my data by during certain point third trial, I will be taking blood samples that I will recollect and then I will analyze it by the enzyme immunosomal rate. And also I'm looking at other factors like their daily food intake and just their overall weight gain. If there's a decrease in this, it will show that they are still non-dead like signals. And also I'm going to look at any specific behaviors that could be known as potential indicators of nausea. Since pregnant mice, 6 mice cannot throw up, it's going to know any behaviors, and I'll be recording it through video. So my anticipated outcomes is that I will see an increase in the activity and serum level of GDF-15 and post-growth treated mice. And looking at my graph, this is not real accurate number. This will just give me an overview for what I expect to happen. So I just see all the experimental groups, they had a decrease in their GDF-15 levels, while the control group still had overall high elevated levels. And also, I expect to see a reduction in the nausea and all these symptoms and consequent bad treated pregnant mice. And lastly, I will expect to identify the most effective and efficient second dosing with administered consequent map. I believe that most efficient dosing will be the medium dose since it's not too high to where it will cause a very drastic change in the GDF-15 levels. And also, I don't think it would be good for the low dose group will still cause the level to be exactly very high. I'm so paid. And then this would then lead me to my super A2, which would evaluate the overall safety of constant mag by a second effect on maternal health and the fetal developmental outcomes during the animal and pregnant mice. This is very important, especially the incident of the litter line, which was given to pregnant women in treating morning sickness, but had resulted in the B1 of birth defects. So it's important to show that any medication that is going to be used on a pregnant woman that will overall have a good impact on her health. And then I realized that if GD15 actually is neutralized internal blood by the injection of growth brand into pregnant with high levels of GD15, it will have good effects on maternal health and fetal developmental outcomes. So for my methodology, I'm going to be looking at maternal health and the fetus health of my A2. And using the same mind that was in system input, I'm going to be looking at the overall the maternal health and the fetus health. So for maternal health, I'm going to monitor for any sign of distress by adverse reactions, while also performing blood chemistry and organ function tests, moving it up on the heart, digestive organs, the kidney, and the enterocyte to detect any type of toxicity. Then also for the fetal health, I'm going to monitor his growth by using a micro ultrasound, as shown in these images right here and right here. And then I'm going to measure the fetal size while using the micro ultrasound and after it is born to check if that is at the right height and just weight. Then also after that, I'm going to examine for any cognitive abnormalities are developing to the legs. And then lastly, I'm going to take placenta tissue from the mother to determine the placenta overall morphology and function to determine if the fetus was overall healthy while in the placenta. But while in the room, my bad. So my anticipated outcome is that during aftermath of post-growth there will be good changes in maternal health and fetal development outcomes will be there in the post-growth treated pregnant mice. My future study to test the other therapies that I introduced at the beginning to determine which one to most efficient at decreasing DDR15 and reducing non-involuntary consent. Also, I wanted to do more research on etiology of non-loving pregnancy to fully understand the state of the hormones affecting that baby involved. So I just want to thank God for just helping me and giving me the knowledge just to do this. My family who has encouraged me and guided me. And I just want to thank my professors who have been teaching me about all 4 years. And I just want to thank God Master for being my path to the light. And we have our
Good job. And one that we take a lot of issues.
Yeah.
The levels of GDF-15.
Yes.
They vary widely.
Yeah.
Because some of them are severely affected by the warning series. So to one where they keep carrying pregnancy. So that's all true.
Yeah.
What's the effect of women have if they can have children having multiple children? So by child 2, child 3, child 4, what's the effect of this after multiple pregnancies?
So it has like found to be like it's there. It's like for some women they will still like have high levels of it to where they will still experience morning sickness. But some will, after they have it, that like first pregnancy, they won't have it no more. So it just varies all around on it.
Okay.
Crime. And so this problem has been already trying solved before, right?
Yeah.
You have mentioned you come across thalidomide. We've talked about that. Yeah, thalidomide was the first they put in the market, which the biggest problem that they had was enantiomeric form teardrops. So what is the probability that this monoclonal antibody can actually go into an enantiomeric form?
I'm not overall sure, but I definitely would do more research on that. That's why it's very important that I test to see the A2 just to see what it had before we start using it on like human. So I'm definitely going to do more research on that.
Is there a threshold for mice sensing if it is like vomiting? Is it that the mice can't vomit? How would you?
So what I'm gonna do is just like watch like the overall BMG. So like if you're just like sleeping or even if they're just not eating, and that's just sign that you're experiencing nausea thoughts.
But do you think that stretch could be used by infected mice every 2 days?
Yeah, that is true. It could be that too, yeah.
Out there, it can false by inject, which is saline specimen to compare to.
Yeah.
On the bigger status.
Thank you.
And still with such an